Yeah, good evening, Dr. Raj
Good
Bakshi.
evening.
Yeah, good evening.
Good evening, Dr. Sujita. Yeah.
Yeah,
So, Sujita ma'am,
yes, sir.
I have started YouTube
Okay, sir.
and I am very happy to welcome Raj Pakshe from USA.
Yes.
Thank
Actually,
you.
yes, Raj Pakshe name seems to be from Sri Lanka.
Yeah, that's someone famous. Yeah.
So, you are from Sri Lanka, which place?
I'm actually from somewhat smaller place called Kurunegala, but it is closer to Kandy, and I was from University of Peradeniya, which is from Kandy, if you have heard about.
I have visited Peradeniya University 3-4 times.
Oh, actually, that's
Yes.
wonderful to hear.
Correct.
So I must have been here there by the time professor there. So because I've been there from 2010 until 2020. So almost 10 years I was there in University of Peradeniya.
Dr. Nimal Adhikaram was a head professor and head in my biotechnology.
Oh,
Botany,
okay.
botany. He was my friend.
History.
So, Dr. Sujita, you can proceed.
Yes, sir. Okay, sir. Yeah, before proceeding to the main session, just I would like to give the brief introduction about Dr. Sanjeev Dilan Rajpakshe. As an international microbiologist cooperation initiative, Microbiology Society India, we are regularly conducting lecture series from overseas. So today we have our guest speaker, Dr. Sanjita Dilan Rajpakshe. Is working as a graduate teaching assistant in University of Kansas, USA. As an entrepreneur, he created few startup companies where he utilized his R&D and management abilities, and he is also a faculty at several academic institutes, including Postgraduate Institute of Science, University of Virginia, Faculty of Technological Studies, Yuva Varanasi University, and Australian College of Business and Technology. His research interests are chemical synthesis, material science, physical chemistry, nanofabrication, and nanocomposites, spectroscopy, molecular biology, and biochemistry. He's having several research publications, book chapters, conference presentations at several countries, and other research communications. So it's really a glad to have you, Dr. Sanjita Dilani Rajpaksha, as today's speaker. So over to you, sir. Please start your session. I request you to start the session.
Okay.
Raj Pakshe is a co-host. He can share the slides.
Oh yeah. I'll share my screen. All right, so I hope everybody can see my presentation, the first slide.
Yes.
Okay, so let's start and let's move on. So I believe it's good evening for you guys, even though it's pretty much good. It's the morning for us. So good evening, guys. So I'm going to talk about the antimicrobial nanomaterials, particularly the future of these nanomaterials and what we can do and what would happen to these nanomaterials and what are the regulatory and so on. So I'll give you a brief overview about what I'm going to talk about throughout this session. So obviously it's about nanomaterials and specifically antimicrobial nanomaterials. And I'll talk about how they do these things, how they actually kill these microbes, or actually they inhibit their growth, and how they process likewise. And then the term broad spectrum activity, the actually why these nanomaterials are specific, the specialty of these nanomaterials, that will be spoken with the next topics like the broad spectrum activity and the reduced resistant development. I guess you can hear me.
Yeah, it's audible. Please continue.
Okay. So I was just checking. So then I'll be talking about the combination therapies, what we can do with these nanomaterials and how we can incorporate other things with these nanomaterials and proceed on, and how to minimize or how to minimize the risk, or how to how to overcome the so-called problems with the nanomaterials with these combination therapies, and then how to synthesize these nanomaterials and the applications, and then the most special important things, the safety and the regulations and the future aspects. So when it comes to nanomaterials, I believe you all know, I mean, by that point, you all must know, might know about nanomaterials. At least you have heard about the word nano. So it's about tiny things which are in the range of 1 to 100 nanometers. So 1 nanometer is a 1 billionth of a meter, so it's that small. So if we can fabricate, or if we can manipulate any matter that within that range, we call them nanomaterials, and they exhibit unique properties like more specific physical, chemical, and biological properties that are different from their bulk materials, and these properties are much important and much specific, and we can use these properties to make special things. And these things can I mean, there are different types of nanomaterials like nanoparticles, nanotubes, nanowires, and nanocomposites, quantum dots, and so on. So, and these things can be composed with different types of substances like metals, metal oxides, and semiconductors, polymer nanomaterials, carbon-based things, and they possess specific abilities that are different from their bulk materials, and specifically the surface to ratio volume is high whenever they are becoming smaller, and that is the most important aspect about these nanomaterials. And when it comes to the antimicrobial nanomaterials, they are actually, these materials originally have the antimicrobial property, or they inherit the antimicrobial property. But what we do is we enhance the property, or we give some special ability to these already active materials. I mean, these materials already have the antimicrobial ability. Like, for example, the metallic nanomaterials, like metallic materials like silver, copper, or gold, they have the antimicrobial property. And when it comes to the nanoscale, they have special antimicrobial properties and enhanced microbial properties. And we can have other metallic nanoparticles. We can have other carbon-based, like graphene, and carbon nanotube-based antimicrobial nanomaterials. And then we can have metal oxide-based antimicrobial nanomaterials like zinc oxide, platinum dioxide, hydroxyapatite, magnesium oxide, likewise. And there are several other polymeric nanomaterials as well that have these antimicrobial ability. And there are these lipid-based nanomaterials and also other nanomaterials. So how they do this thing, I mean, how they kill the microbes, how they inhibit their growth is basically depending on physical properties. They are physical activities, and most well-known processes are passive processes. That means they do not incorporate any sort of energy or anything. So like physical interactions, like these nanomaterials can physically interact microbial cell membranes or microbial cell components, or they can disintegrate them, or they can rupture them, they can cut them technically, and they would lead the ultimate death of the microbial cell. And then and also they can release cationic things, so they can exchange metal cations. So likewise, they can disrupt the cell processes, and also they can make reactive oxygen species, which can interrupt the cell membrane and destroy them. So these are the passive or the well-known antimicrobial processes of these nanoparticles. And the first, I mean, after that, we have these activated processes, where we incorporate any sort of some sort of energy, like light activated antimicrobial nanomaterials, where we have two types of nanomaterials, like photocatalytic antimicrobial nanomaterials and photothermal antimicrobial nanomaterials. So the photocatalytic, and I mean, in this slide, you can see some of the nanomaterials that we prepared through in following years. I mean, they are, some of them are titanium dioxide nanomaterials, some of them are zinc oxide nanomaterials. So they inherit the the antimicrobial property, and they can kill microbes using the photocatalytic process. So what they do is they absorb light, some sort of specific light, and that light would excite an electron, and this excited electron can react with atmospheric oxygen and make free radicals, and these free radicals react with atmospheric the water, that means moisture, and they make reactive free radicals, reactive hydroxyl free radicals, and these free radicals can disintegrate any sort of microbial cell component, or basically organic component, and also they can basically release metal ions and kill these microbes. So, and once they are in the microbial cells, so they can further disintegrate the nucleic acids and other cell components. Likewise, they would destroy the microbial cell membranes, cells. So that is how these photocatalytic antimicrobial nanomaterials works and kill the microbes. And the second types of light activated nanomaterials are the photothermal antimicrobial nanomaterials. The difference is they generate heat upon specific light radiation, because of the localized surface plasmon resonance. Due to the high frequency plasmon resonance, they generate heat, and this heat would lead to the ultimate damage of the microbial cells. And after that, the second type of energy activated type of antimicrobial nanomaterials are magnetic activated antimicrobial nanomaterials, And these magnetic activated antimicrobial nanomaterials can be further categorized into two major groups, like magnetophysical antimicrobial nanomaterials and magnetic hypothermia of antimicrobial nanomaterials. So here as well, you can see two types of SEM images of two types of magnetic activated nanomaterials that we prepared with a collaboration of a private industry. So these are magnetite nanomaterials. That means they are iron oxide, basically iron oxide, a type of iron oxide nanomaterials, and they inherit the magnetic property, and we can use this magnetic property to localize healing of nanomaterials or targeted delivery of the nanomaterials to specific areas or specific pathogenic places, and then we can activate these nanomaterials. There are two types, as I said, there are two types: magnetophysical and magnetic hypothermia. The first one, the magnetic physical antimicrobial nanomaterials, I mean, they have the magnetic property. I mean, they are magnetic materials. So we can use a magnetic field to basically move them around to somewhere we want. So actually, we can use them to target delivery these nanomaterials, and then we can manipulate them in that specific certain areas. So it enhances the efficacy, and it minimizes the potential side effects. And also, since they have the since we can manipulate these nanomaterials, since we can move them around using a magnetic field, we can recover them after the processes that we need, that we have done. After that, we have the second type of magnetic activated antimicrobial nanomaterials that are magnetohypothermia antimicrobial nanomaterials. So they already have the magnetic property, so we can use a magnetic field to move them around to wherever we want. And on top of that, by using an alternating magnetic field, we can create heat. We can generate heat using these magnetic nanomaterials. So what we do is we can use the magnetic field to move them around to a place, to a certain area in the body, let's say a certain infected area. And then after that, we can use an alternative magnetic field to activate these nanomaterials. Then we can generate heat, and then we can kill these pathogenic substances or pathogenic areas. So likewise, we can selectively kill them. We can deliver them in a targeted manner, and we can offer several potential advantages, like enhance the targeting, and we can have other synergetic effect, like we can enhance the antimicrobial ability, and also like in many cases, like you already know that in many cases of infectious diseases inside the body, like after a surgery maybe, we have to give antibiotics or huge doses of antibiotics, also things like that. But we can use these targeted delivered magnetic nanomaterials to deliver them to the specific places, and use very small amounts to do this certain thing. And by doing so, we can reduce the resistance, and we can control release them. And the most important thing of these nanomaterials are that they can kill wide range of microbes, wide range of pathogens like viruses, bacteria or fungi, or maybe drug resistant microbes like penicillin resistant uh, RSA, MRSA. So likewise, we can they are versatile. We can use single type of nanomaterial to kill a vast range of microbes, unless using a specific antibiotic to kill a specific pathogen. So that is one important parameter of these nanomaterials, the broad spectrum activity. So they can effectively target a wide range of microorganisms, and it would be valuable in the face of emerging antibiotic resistant bacteria, and most importantly in viral outbreaks like we had in past few years. Why they can do this broad spectrum activity? That is because they destroy these microbes, or they kill these microbes in a different manner than the antibiotics does, because they mostly do this in a mechanical manner. So microbes wouldn't be able to adapt that easier for these material. So because of that, the resistance would be low. So the microbial resistance is a kind of a headache to the global population. It's a kind of a number one case issue. So we can address those sort of issues with these nanomaterials. So, for example, like silver nanomaterials, like they disrupt the cellular membrane, they interfere with the metabolic processes. So, copper nanomaterials, they penetrate cell membrane and they generate reactive oxygen species. Zinc oxide nanoparticles do the same thing. Titanium dioxide nanomaterials, they produce reactive oxygen species. And so like nanomaterials destroy these microbes in a different manner than the antibiotics does. That is more like a mechanical manner. So because of that, as I said, these microbes wouldn't be that easier to adapt to the antibacterial nanomaterials. So the resistance would be low, as I said. And that is because these nanomaterials have multiple modes of action in different to the antibiotics, and they have enhanced properties, and they have reduced targeted specificity, and we can incorporate several other things to these nanomaterials, several other already antibiotic active materials to these nanomaterials, and we can have synergetic effects. And the mechanism of action is different, and most importantly, surface to volume ratio is high. So there would be a high surface, there would be a high area for these material to interact with the microbes, and they can efficiently kill these microbes, and also they would provide enhanced surface area for the antibiotics or other reagents if we attach them onto, and we would need very small amounts of these other antibiotics or antimicrobial agents to incorporate. So that would also lead to the reduced microbial resistance development. And that is the most that is somewhat specific about these nanomaterials, the combination therapies. That means we can add traditional antibiotics or other natural antimicrobial agents onto these nanomaterials. By doing so, we can enhance the antimicrobial activity of these materials. So, like we one time we like in this images you can see the cotton gauze incorporated with zinc oxide nanomaterial modified antibiotic attached zinc oxide nanomaterial modified cotton gauze that we used, and we were able to minimize the concentration of the antibiotic dosage by 100-fold, and we could get the same antibiotic effect or enhancing antibiotic effect using these materials. So we incorporated them with other textiles, other fabrics as well, and we use them as tropical antibiotic gauzes that that we used on diabetic ulcer patients. So we were able to cure these patients with these antibiotic impregnated cotton gauzes, and you can see the CM images of these zinc oxide nanomaterials that have sharp edges. And that is how they kill the microbes that or they disintegrate the cell membrane. So we were able to incorporate these sort of combination therapies to several other industrial fabrics as well. So that's all about how these nanomaterials are important, how they do the specific thing, specific antimicrobial activities, and how they kill the microbes. So when it's about how these nanomaterials are formed, how can we synthesize them? So there are two basic methods, top-down and bottom-up approaches to manufacture these nanomaterials or synthesize these nanomaterials. So the top-down method, basically we can have a bulk and we can cut it down, we can break it down to until we are having the specific shaped nanomaterials, or we can have the bottom-up approach where we add molecules or atoms together to until we are having the specific nanomaterials. So there can be three different we can have all three different approaches of physical, chemical, or biological methods to produce these nanomaterials. So physical methods are mostly top down, like we use a bulk metal or something, and we can use a laser ablation or electron beam lithography or mechanical grinding, milling sort of thing, or to bring it down to the nano level. And we can have the chemical approaches where some of them are top down, where we can use an anisotropic dissolution, or most of them are bottom up, where we use tiny molecules or atoms to produce these nanomaterials, like precipitation methods, salt gel methods, pyrolysis methods, and chemical reduction methods, and so on. And also we can use biological methods, where we can use plant extracts or bacterial or biogenesis extracts, fungal, algae extracts to produce these nanomaterials. Because certain microbes already make nanomaterials inside their bodies, so we can extract them and to use them in other applications. So here you can see some of the nanomaterials that I produced throughout the past few years, most of them are in different shapes, like spines, shapes, flakes, and they have ridges and spherical nanomaterials, and we incorporated it. We could incorporate them in many surfaces. I mean, almost any surface: plastic, glass, metal, fabric, paper, anything. So, I mean, you're gonna have to have the correct method to incorporate them and to attach them onto certain surfaces, or to apply them in certain applications. By doing so, we can use them in creating probably the best shield against these pathogenic microbes. So doing so, I'll move on to the applications of these antimicrobial nanomaterials. Basically, I'll talk about biomedical applications, the antimicrobial coatings, and how we can use them as wound dressings and sutures, and how we can use them in implants and tissue engineering, the antimicrobial textile fabrications, and how they can be used in environmental and water purification applications, and food packaging preservation methods. So in biomedical applications, we can use them in several processes like drug delivery systems. We can use them in imaging as imaging agents, like in MRI imaging, and we can use them in tissue engineering. We can modify these nanomaterials to sense certain pathogens or certain microbes or certain biomolecules, and we can use them in cancer therapeutics, and basically mostly we can use them in wound healing and bioimaging techniques. We can use them in incorporated with frozen materials to in imaging techniques, and here I have used some of the drug delivery zinc oxide nanomaterials that we created using cisplatin, the anti-cancer drug delivery nanomaterials that we created with zinc oxide nanomaterials. So they were targetically, we could deliver them to certain places and release the drugs. And when it's come to the antimicrobial coatings, it is much important, because we can use them in many areas. Because I mean, like last couple of years, we were damaging with a certain viral attack. So in those sort of scenarios, we can use these nanomaterials in creating important shield against these pathogenic bacteria. So these coatings are thin layers of nanomaterials that inherit the antimicrobial ability, and they can provide prolonged protections against the growth and spread of microorganisms, because we can incorporate them on any surface, and they would attach them onto the surface. And most of these nanomaterials are like minerals and metals, so they wouldn't undergo much change throughout the time. So they would they would be active throughout the time that they are on those certain surface, and also they can reduce the risk of infection and the spread of diseases. So we can incorporate them in various coating materials like paints, plastics, ceramics, textiles, and medical devices. So we prepared several these nanomaterials with zinc oxide nanomaterials and iron oxide nanomaterials, and we incorporate them in several coating materials, surface treating materials, and we incorporate them in paint, and we use them in hospitals in past COVID-19 outbreak, and they gave us remarkable results. And I mean, they help us in a remarkable manner to fight against these pathogenic viral outbreaks and other microbial infections. And we can, importantly, we can use them in a wide range of places like healthcare offices and public places, and they are not toxic compared to the most of the antimicrobial agents that we use in nowadays. And I will talk about why they are important and how they are not toxic compared to the other things that we use. And then the next application is the wound dressings, and in sutures we can use these nanomaterials. And as I said earlier, we incorporated them in cotton gauzes. And as you can see in here, you'd see the inhibition zones again around these nanomaterial incorporated fabrics. And we tested them against many bacterial, viral, and fungal strains. Like we tested against these gram-positive and gram-negative bacterial species like Staphylococcus aureus and Escherichia coli. And we tested against viral species like corona virus as well. And these wound dressings can enhance the healing, and they can be applied in tropical manner, and they can participate in moisture management as well. They I mean, we can modify these nanomaterials to maintain the optimum moisture around these wounds, and that is much important. And we can use them as a scaffold structure. And when it comes to sutures, you know that sometimes sutures can get infected and make all sort of damages, and we can use these we can incorporate these nanomaterials into these sutures and make antibacterial sutures, and we can use them as drug delivery materials. And next application is we had used them in implants, in biological implants and tissue engineering. So we can modify some of the biological implants, bone implants, with these antimicrobial nanomaterials, and they can reduce the risk of infection. Like we can, like people use titanium rods and those biological implants made with metals like titanium and other alloys. So we can modify them with these nanomaterials, and they can enhance the functionality of these implants, and they can most of the times, if we after surgery of the after implantation surgery, people have to undergo through a high dosage of antibiotics. Instead of that, we can use a localized delivery. We can incorporate the antibiotic onto the nanomaterials that are attached to these implantations, and we can produce localized antimicrobial effect. And some of these nanomaterials, and some of these shapes of these nanomaterials can promote tissue regeneration, and we can have better attachment onto the tissues. So you can see some of the CM images of these titanium dioxide and magnesium oxide nanomaterials that we used on biological implants. And next thing is on textile and fabrics. So we prepared many nanomaterials with several semiconductor nanomaterials, and we incorporated with them fabric, several types of fabrics in commercial scale. I mean, we made like 100 meters or 1,000 meters of these fabrics using these nanomaterials, and they can be used as antimicrobial textiles, or they can be used to make specific fabrics or garments that are used in healthcare facilities that have the antimicrobial ability, and they can they can be used to reduce the risk of infection. And we can use these textiles as hygienic and odor control materials. And by using correct methods of applications and correct types of chemicals, we can have better attachment of these nanomaterials onto these fabrics. Like in these SEM images, you can see how we incorporated these things onto the I'll get the so here you can see how we have incorporated these nanomaterials onto the fibers, and how they can activate as nanomaterials while they are on these fabrics. Okay, so the next application is environmental and water purification processes. We can use them in many environmental applications, like water desinfection, in water filtration, as antimicrobial coatings, as wastewater treatment materials, soil remediation techniques, and air purification techniques. So some of these nanomaterials that we prepared, like these magnetite iron oxide nanomaterials and hematite iron oxide nanomaterials, they can be used as water disinfection nanomaterials. So what we basically do is we produce these nanomaterials and we attach them onto specific surfaces, and we can use them in water purification or water disinfection whenever microbes or pathogen microbes go through these nanomaterial attached membranes. They kill them and do the disinfection. And they can be used as for water filtration materials, like the iron oxide nanomaterials that we prepared. We used them as heavy metal absorbing nanomaterials, and since they have high surface area, they can effectively absorb these microbes and other debris or other heavy metals, and they would do the water filtration. And the third type is antibacterial coatings. We can incorporate them in any type of surface and use them as antibacterial coating. And we used titanium dioxide nanomaterials as wastewater treatment techniques materials. Importantly, we used there was a project in Andhra in India, in Brandix Aprocity, and where we used titanium dioxide materials as wastewater treatment materials. So we were trying to use nanomaterials in bulk scale for wastewater treatment facilities, because we can activate them with sunlight, and we can use them to disintegrate inorganic or organic matter. And we can use them as soil remediation materials, where we can use them in cultivating lands in specific manner to absorb heavy metals and other toxic materials that we throw into the environment in these farming lands. And we can use these nanomaterials incorporated with other fibrous membrane, and we can use them in air purification applications. Likewise, we can use these nanomaterials in many of environmental and water purification applications. And next thing is that we can use them in food packaging and preservation applications. So where we can use them in packaging materials, and these packaging materials would provide the antimicrobial activity. They would act against these pathogenic bacteria, pathogenic fungi, or pathogenic virus. And because of that, they would extend the shelf life of the food products, and they would reduce the risk of foodborne illnesses. We can control release these some of the antibiotics to the packagings, or antimicrobial agents to the packagings, like antimicrobial. There are antimicrobial essential oils that we can incorporate onto these nanomaterials, and we can then attach them onto the food packaging, and then they would act as slow releasing or control releasing antimicrobial agents. And we can have them in active packaging and as surface coatings in delivery processes, and we can use them as edible coatings as well. And we can use nano films or nanocomposite films to produce these commercial scale of packaging materials. So all these are applications, and we talk about all the good things about nanomaterials and how can we use them in against these pathogenic things and all. But there is a certain concern about these nanomaterials, and people are thinking about whether they are giving the proper thing, or whether they are making any matter on global population or not. So because of that, we have to concern about the safety and how to regulate these nanomaterials. And when it comes to the safety and regulation, we have to talk about the toxicity and the risk management, and the environmental impact of these nanomaterials, and how to standardize these nanomaterials, and how to test these nanomaterials, so the testing protocols and all, and what are the regulations and the guidelines. So first, the toxicity and the risks assessment. So there can be toxicities that are incorporated with these nanomaterials, and when it's come to the toxicities, these toxicity levels are much minimum compared to the much of the household items that we use in today's life, even though we use them in a regular basis. But yeah, obviously we can inhale them, we can have them on skin, so sometimes they can make allergic reactions or so on. And we can accidentally eat them, or they can have an environmental impact. They can we release them, ultimately we release them onto the environment, to the water resources, and to the aquatic areas. So they can be damaging these environmental arenas. So we have to concern about these things as well. So we have to think about the hazard identification, how much hazardous are these nanomaterials, and what is the dosages of the when it comes to the hazardous of these nanomaterials, and how we exposure those ourselves to these nanomaterials. So the exposure assessment, and like I said, the dose response assessment, how much dose is responsive to how much damage, likewise, and what is the risk, and how to characterize that risk. So and then next safety concern is the environmental impact. We release them into the environment, and anything that we release in the environment by not by mean of the environment can be toxic or can make a harm to the natural balance. So we have to regulate the release. We have to have some sort of a standard again on the releasing these nanomaterials to the environment, and we have to think about the ecotoxicity. They can be toxic to the aquatic life forms, and they can be bioaccumulative. They can accumulate throughout the plants or animal chains, and also throughout the microbial chains as well. So they can have a long-term behavior, since they are, most of them are semiconductor materials, or most of them are minerals or metals, and they wouldn't undergo much of the change, so they would remain much same throughout the time, and they can arise water and soil contaminations as well. So we have to have a standardization. We have to have a testing protocol against these materials. So first we have to standardize. We have to think about the toxicity, the risk, the compatibility of these nanomaterials against biological systems. So if we think about these nanomaterials, many of them are already in our environment, like semiconductor materials. Like you can already find zinc oxide or titanium dioxide or iron oxide materials or ceramic materials that are in environment, that are naturally occurring in environment as minerals or other things or other composites. So they are already in our environment, but we have to assess the toxicity of these things. So we, by ourselves, we did the characterization of the toxicity of some of these nanomaterials, and we used several high dosages of these nanomaterials, and we tested them against live animals, laboratory animals, and other cellular systems as well. And we found that they are not toxic, and they are perfectly okay to be used in biological systems, and they are perfectly fine, and they are perfectly compatible, non-toxic. So most of the concerns that people are thinking can be regulated using the right dosage or the right type of the nanomaterial. And we have to we can characterize them using several different techniques like CM, TEM, X-ray refractive index diffraction method, and dynamic light scattering, DLS, Fourier transmitted IR. Likewise, we can use them to see what are these nanomaterials and shape, the size, and all sort of things. And we can assess the antimicrobial activity of these nanomaterials, use several different techniques like MIC, MBC, or zone of inhibition assays, where we see the inhibition zone, or how much time it takes to kill assays, the time kill assays. And we can use several different cytotoxicity and biocompatibility measurements like in vitro and in vivo assays to measure the toxicity and the biocompatibility of these nanomaterials. And we can use several different parameters like temperature, pH, the resistance to degrade, and the maintenance of the antimicrobial properties to measure the stability and the durability of these nanomaterials, and then we can make regulatory compliances, and doing so we can make a standardization against these nanomaterials, and we can use them in commercial aspects. And finally, we have to regulate, and we have to have proper guidelines against these nanomaterials, because we already use many toxics, many non-harmful chemicals in everyday life, in everyday farming or everyday industrial processes, and we already release them into the environmental systems. But what we do is we regulate that. By doing so, we can maintain the proper balance in our environment and in our global population as well. So we have to have regulatory authorities, like in several countries, they have their own bodies, like in USA, they have FDA or EPA, or in Europe, they have ECHA, likewise. And we can have safety assessments, we can do assessments against these nanomaterials, we can check actually whether they are toxic or not, whether they are compatible or not, and we can register them, and we can make notifications whenever somebody make a nanomaterial, whenever somebody invent something, we can do the safety assessment and register them, and we can make a database of sort of a thing, and we can make a labeling and product claims, and we can do the ultimate standardization. And by thinking about the environmental considerations and intellectual property protections, we can actually make a stable system for these nanomaterials to be used against these pathogenic microbes. So even though we can use these nanomaterials in several aspects in day-to-day life and in commercial arenas or in other aspects, there are challenges like the scalability and the cost effectiveness, how much we can scale it up, whether we can use them in industrial arena or not, and whether it is cost effective or not, and whether they are biocompatible and can we use them in long-term aspects, and we have to have the regulatory frameworks. So we actually throughout the past years, we produced these nanomaterials, we tested these the compatibility and the toxicities, and we produced them in several commercial arenas, in commercial scale, in bulk scale. So they are industrially viable, and these processes are industrially viable, and they are much cost effective. And since we have to use very small amount of these nanomaterials, there we can use very small amount of raw materials, and we can use them in targeted delivery systems and diagnostic applications as well. Even though there can be safety concerns, we can use them in targeted manner, and we can kill the specific things as only, and we can use them in combination therapeutics, and we can make other imaging techniques as well. So yeah, we can produce these nanomaterials, and also people think these nanomaterials can make safety concerns. So, but what I suggest is that so there is a pathogenic wall that can make harmful matters against humans, and there is a weapon with us that's called these antimicrobial nanomaterials. We know that it can be harmful, but in a regulated manner, we can use them against these pathogens. So as a weapon that we use in a war, we can use it to kill our enemy, not to kill ourselves. So by understanding these nanomaterials, and by using them in proper applications, we can get the maximum out of these nanomaterials. So that's it about today. So thank you very much for your cooperation and all. So I thank very much for the Microbiologists Association of India for your time and for your opportunity that you given to me. So here as well, I use two images that are important to me. One is Sigiriya. It's a ancient capital in Sri Lanka, and here they have used, even though they might have known or they might have not known, they have used many nanomaterials in paintings, in medicine, and in other various sort of applications. And here as well, I mean, University of Kansas, where we use these nanofabrication techniques, the Center for Nanofabrication, to manufacture these nanomaterials in future aspects. So then again, I thank you all for your valuable time, and I'll hand this over to Dr. Sujita for the proceedings.
Thank you so much, Dr. Dilan Rajbakshe. It was really a wonderful and in-depth illustration of nanotechnology and nanomaterials as antimicrobial agents. And I hope that this will be very much helpful for all of our students and faculty and research scholars. Now the session is open for discussion, and if any participants is having any question or anything, you can ask Dr. Dilan Rajbakshe.
Hello. Am I audible, ma'am?
Yeah, Dr. Shravani. Yeah, please. Yeah.
Can I ask a question?
Yeah, please.
Sir, as you have discussed 3 methods for the synthesis of nanoparticles, they were physical, chemical, and biological method. But in some of the research paper which I have read, in biological method, they are making use of NaOH also. So does it make any difference, or does it affect the results? What I mean by the result is the final synthesized product with respect to shape and size. So is there any, does it affect as we are using NaOH, or any alkaline conditions or pH has to be maintained?
Hello, Rajpakshe sir, question is to you.
Yeah, actually.
Where is Rajpakshe?
Dr. Rajpakshe sir, please unmute it. Yeah.
So it doesn't actually matter. I mean, in various manufacturing processes, we use sodium hydroxide, and sodium hydroxide itself we can use to synthesize the oxides and other things. So it doesn't matter, but I mean, most important thing is that after the fabrication of this nanomaterial, that they have to be in a neutral condition. So we have to maintain the neutral condition. So if the neutral condition affect the nanomaterial, then we have to use capping agents or tapping agent to stabilize these nanomaterials. So that's it. Other than that, using sodium hydroxide wouldn't be that important. I mean, wouldn't be that different. Thank you.
Okay, thank you, sir. Thank you so much.
Yeah, thank you so much for everyone. Since I'm actively
Hello.
Yeah, Aruna ma'am.
Hi. I'm Dr. Arunima Biswas from West Bengal. So may I ask a couple of questions?
Yeah, please carry on.
Yeah, sure. Yeah,
please.
Okay. First of all, excellent talk. Excellent, just excellent. Thank you so much. So I have a couple of questions, which is like when we are using these nanoparticles in therapeutics, how do they interact with our immune system? I mean, in terms of both immune stimulation or immune toxicity. That is one question. And another question is, how do they evade, our system of xenobiotic metabolism, where often this system flushes out particles they think they are not good for the system. So these are the couple of questions that I have.
Yeah, thank you. That are so important questions. So, yeah, so we tested against some of the, epithelial cells and epithelial lung cells and all. Like, we tried them on mammalian cells, and we saw that they wouldn't make any toxic matter or any incompatibility. So people say that there is a safety concern about the immunity and how would it affect on the immunity, but these nanomaterials react mostly in a physical manner rather than a biological or chemical manner. So it wouldn't much affect on the immune system. Immune system itself wouldn't identify these nanomaterials or identify these nanomaterials or as antibiotics or any other immune boosting or immune reducing thing. So because of that, according to my experience, they wouldn't make much effect on the immune system or the balance of the immune system. So that's my understanding, but obviously we have to do further investigations on those, because there are very less literature on the effect of the immune system and the immunity of the mammalians, most importantly. So we have to do that. And so when it's come to the bio processes that they eliminate naturally, so there are certain processes that some of these nanomaterials can be eliminated. Like it basically depends on the type of the nanomaterials, and we have to think about whether there is a process that there's already biological process that can eliminate those things. So most of the times we can use these specific nanomaterials that can be used in biological eliminative processes, so that can minimize the harm if it makes anything. So likewise, it's that for certain applications, we have to choose what is the most appropriate nanomaterial. That is the most crucial thing. There are thousands of nanomaterials and there are thousands of shapes, and when it comes to the shapes and size of these different nanomaterials, and they would become entirely completely different. So it's hard to say this nanomaterial is that compatible or toxic or bioprocessive or not. So we have to manufacture them, we have to create it, and we have to try them on the biological process also, and then after only we can decide whether or not to use them in certain applications or not. Thank you.
Okay, thank you so much. Thank you.
Yeah, thank you. And in last, I would like to ask one question, Dr. Rajpakshe, because I am working, we have in our recent work, we have prepared biofabricated nanomaterials for diabetic foot ulcers. So have you tested this, your material as anti-biofilm agents? So is that?
What was the nanomaterial that you were
We
using?
have used, biofabrication. So plant materials we have used.
Okay.
So, yeah, we have capped it.
Yeah. I have prepared these zero-valent ion nanomaterials using tea extract, that the tea tree extracts. So it's a kind of a biological process, and we use them in as microbial agents, and then we tested their compatibilities and toxicities. So as far as I know, we didn't find any toxicity. And we did immunoassays, we did histopathological assays as well, using lab rats, and also, but to my knowledge, I didn't find any toxicity to many of these nanomaterials. But still, I'm also trying to see any difference. But the thing is that, let's say, for example, zinc oxide, the daily dosage that we can take zinc oxide is about 40 milligram. So
Yeah.
that's a huge amount. So even though we use these nanomaterials in these applications, we use very, very little amount of these nanomaterials. So I don't think that would make any toxic effect. So that's what I think. So, but still, we have to investigate further about this nanomaterial.
Yeah. Yeah, thank you so much. So with this one, we'll end this session. So thank you very much for all of you. And with this, we'll close the session. Thank you very much, for Dr. Rajpakshe, once again. Yeah, thank you.
Yes,
Thank you.
really
Yeah.
excellent session.
Yes, sir.
But I will request you, Sujita ma'am,
Yes, sir.
you
Yes, sir.
take his WhatsApp number and add his number in our international group.
Sure, sir. Sure, sir.
So
I'll
other teachers also will get benefit.
sure, sir. Sure, sir.
Okay.
Yes.
And thank you, Rajpakshe.
It's a privilege for
Yes.
talking
And
to you guys. So thank you very much. Thank you all.
sometime if you happen to visit India,
Yeah, sure.
please call me, tell me. Definitely, I will arrange your lectures in various universities in India.
Yeah, thank you. Thank you very much. I've been to India in several times, so, and I'm hoping to come to India in future as well. So I'll definitely contact you guys, and I'll
Yeah.
be in touch with you guys. Thank you.
Sure, surely. And I thank you, our Microbiology Society President, Dr. A.M. Deshmukh sir, for creating such a wonderful platform for international cooperation and exchange of expertism. And really, we are very much thankful to our president. Yeah. So with this one,
Thank you.
yeah,
Yes.
yeah.
So this link will be circulated, and the link tomorrow will be given to even Rajpaksha sir.
Okay, sir.
Thank you. All right. Thank
Okay,
you.
thank you.
Thank you.
Thank you, sir.
So now, Sujita ma'am, you can end the